All right, so we're going to look at some neurotransmitters here, and I titled this "Improving Patient Outcomes." And I think that, obviously we want to try to have our patients with the best, have them come out with the best outcome. But I think in looking at this, we have to figure out an easy way to incorporate this into our consultations with our patients and try to figure out what are key things that we're hearing, so we'll know how to further direct what treatment interventions we have related to their neurotransmitters. And let me say this as I'm getting going here. By the time I get through with this presentation, every one of you in here are going to go, "That's me." One part of what I say in here, you're going to think, "That's me." Every patient walking through our door has some degree of neurotransmitter imbalance, all of them. Okay? It just really boils down to, is that imbalance significant enough to cause some type of clinical symptomatology that we need to do something about, and that we need to address? So, just kind of take that as is as we go through this. So the things I want to talk to you about are, what are the roles of the neurotransmitters. So we'll really define that so we have a base kind of what the significance of looking at these are. Where are the neurotransmitters produced? I've got some good illustrations in here for you so you can start isolating that into various brain regions. And also I wanted to go into that so that when you start hearing information out there about neurotransmitters and assessment methods about neurotransmitters, you can differentiate the information you're hearing and make a scientific valid decision about how you want to implement various assessment methods. Okay, and you'll see where I'm going with that. We'll look at some of the emotions and the clinical findings related to the neurotransmitters. We'll touch on the assessment methods and how to keep those pretty simple and get the maximum information. As much information as we can get back and minimize the cost to the patient. That's what we're looking to try to do, right? How to support them in some of the factors that affect neurotransmitter metabolism. And that one's pretty key, because that last section we're going to get into with those factors that affect the neurotransmitter metabolism, those are the things that typically will get overlooked. And if you just put a patient on, say, like dopamine support, and that's all you're giving them, and you're not taking into consideration the things that influence dopamine support, your outcome is probably not going to be as ideal as you'd like for it to be. So let's go with the textbook definition on exactly what neurotransmitters are, and then we'll put it into a more tangible definition here. They're endogenous chemicals that transmit signals from neuron to a target cell across the synapse. So in short, the way I think about neurotransmitters is they're chemical messengers. They're saying one thing to the tissue that they're trying to interact on, whether that's the next neuron, whether it's a muscle tissue, end organ, whatever the tissue is, they're transmitting some message to that tissue, right? So here's a schematic of exactly how that takes place with regards to neurons. So you're looking at your action potential is traveling down these axons here, and you go from neuron A over to neuron B, and at the synaptic cleft here, you've got release of the neurotransmitter through these vesicles interacting on these postsynaptic receptor sites. Now, when you're talking about these neurotransmitters, they can be broken down into various categories, and these first two groupings are the ones that we think about most often. You've got your amino acids, so your structures such as GABA, glutamate, aspartate there, and then you've got your monoamine. And you may be familiar with hearing about the medications, your monoamine oxidase inhibitors, these types of things. The mechanism on those medications is that they're breaking down the actual neurotransmitter, or they're inhibiting the breakdown of the neurotransmitter itself. But the main ones you're going to look at there are going to be your dopamine and your serotonin. But things such as norepinephrine or your noradrenaline and your epinephrine adrenaline, they have relevance too. And really, when you're looking at these pathways, and I have a diagram of this for you, it really is just a continuation of dopamine to norepinephrine to epinephrine. So you're looking at molecules that are all basically similar to each other. But their amino acids is the key point. The reason I wanted to differentiate that for you is that, and even if you look at the next category with the peptides, which we don't talk a significant amount about in just day-to-day clinical practice, but if you look at all four of those are all going to be protein-based structures. So if you just step back for a second and think, well, how does diet play into this? Well, there's one of your mechanisms by which diet plays into it, making sure that there's adequate delivery of amino acids. And then the ones that don't actually fit that protein base so nicely are going to be your things like acetylcholine. You know, if you break down acetylcholine and think about where the individual components of that are going to be coming from, choline is going to be found more from your food sources and your fats. Things like eggs have a high concentration of choline in them. And then you're also going to have, if you look at the acetyl portion on that, looking at the production of acetyl-CoA, that's a B5 dependent process. So that's going to originate from B5. Actually, the same precursors that are used to flow into your steroid hormone pathways are also going to be the acetyl-CoA that moves into your steroid hormone pathway. It also moves into the production of acetylcholine as well. And ironically on this, if you think about things, and this is kind of where you would want to start in your clinical evaluation of a patient, just look at the patient and as you're listening to them, think, is this patient too excited, or do they not have enough inhibition, or are they too inhibited and don't have enough excitatory activity? So if you think about things in terms of excitatory versus inhibitory, you got to find a balance there. Now, of all these neurotransmitters, the only one that is technically defined as an inhibitory neurotransmitter is GABA. So GABA has a large role to play if you think about all these other ones up here being excitatory neurotransmitters. That's a lot of burden to bear. And this is just kind of a schematic for you, just to remind you of that balance and to be able to maintain this harmony. You got to think about both of these things. Now, as we go through our day, at various times we're going to have a higher level of excitatory activity, we're going to have a higher level of inhibitory activity, just kind of on a day-to-day basis. You're going to get some subtle shifts in that across the 24-hour cycle and as you're going throughout your day. But it's really when you start seeing a significant skew in one side of this balance or the other is when the patients are going to start manifesting some type of symptomatology. Now, let me use the context of that last slide to lead into this one. How many of you can think of people that just in general seem pretty excitatory all the time? They seem like a rubber ball bouncing off the wall all the time, right?
Having conversations.
Right? And then on the opposite side of that, have you met people that they seem like they need a cattle prod to get going sometimes, right? You've seen both of those types of individuals, right? Okay. The general nature, we all have a general nature about us. And if you look at the basic areas or these basic neurotransmitters, if you think about dopamine, acetylcholine, your GABA, and your serotonin, those four core ones that we tend to try to manipulate most in clinical practice. If you look across your patient population, you can find somebody that meets just about every single one of those criteria that will fit into one of those categories. So you can use that piece of information to your advantage. When you're interacting with a patient, if you've got somebody who is a dopamine type personality, that's more of your top A type people. They're go, 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 go. But that person doesn't want to sit there and listen to you explain biochemistry to them. They just want the facts. Just tell me what I need to know, and that's all I need to know, right? But then if you've got somebody who's more of an acetylcholine person, more analytical, they want the facts. Lay this out for me. Give me every single detail of how I got from where I was to where I am, right? So you can think about that in terms of having patients that are going to follow through with you and modifying your consultation process with them.
Those are NASA engineers.
Right, your doctors, your engineers, those are the type of people that tend to be more dopamine-based individuals. But it is a valuable tool to allow you to communicate with the patient. Now again, just kind of touching on these four core neurotransmitters here, you got your dopamine, acetylcholine, GABA, and serotonin. We used to think about these things as dopamine was associated mostly with frontal lobe, acetylcholine was going to be mostly your parietal lobes, your GABA was going to be more temporal, and your serotonin was more occipital lobe based. Well, we really know now that it's not that nice and clean and cut. These pathways actually have their projections stem into other areas of the brain. They're not just isolated to those regions. So what I've got up here for you as we go through each one of these four neurotransmitters, I've got the core neurotransmitter name up here for you, and then under it I've put what I use to assess if this neurotransmitter should be a concern for me to follow through on. So let me use this as an example. When I want to know about dopamine, I actually ask the person, how's your motivation? Are you Do you feel like you have to get up and push yourself through the day, or do you roll out of the bed and you're ready to tackle your tasks as you go throughout the day? You know, how do you have to push yourself? How's your drive? You can ask them things about their libido, but it all comes back down to motivation, right? So that's kind of the one thing that I use. Every one of these that I put up here, the word that falls under the neurotransmitter, that's the first thing I start asking when I want to know more about that individual category for that patient. So if you're looking at personality traits for your dopamine individual, they're going to be pretty strong-willed. They know what they want. Again, just think about your type A people. Think about most of us that are probably sitting in this room. I mean, we're in the profession we're in for a reason. Tend to be self-confident. We're not, you know, really shy. We tend to probably be outspoken about our opinions about things, that type of stuff. And we're more comfortable with facts and figures. Now, here's the key about this, and this is the reason I wanted to put a personality trait and a deficiency trait up here. If you've got the personality, if you've got someone with a dopamine personality and they've got a dopamine deficiency, that deficiency is significantly affecting them. That's their core nature, and they need support in that core nature. But if you've got someone who's a dopamine personality and yet serotonin is where their core deficiency is at, that neurotransmitter deficiency is in general not probably having quite as big of an effect on them. Does that make sense? Okay. So if you're seeing these types of symptoms, these deficiency symptoms pop up in an individual that has the personality traits consistent for that neurotransmitter, you really got to do something about it, because if you don't, it's absolutely affecting other areas in the body. I'll give you a clinical example on this. If you've got someone who walks in with that personality type and they're having deficiency symptoms, the chances that they're going to be constipated are pretty strong. Because again, think about dopamine as being like a gas pedal. It's a stimulator in the body. It gets things going. That includes our gastrointestinal system. It actually includes everything that we think about as being parasympathetic oriented. Gastrointestinal function, endocrine function, your hepatic activity, that's all going to be it's all going to function at a less than ideal pace in an individual with a dopamine deficiency. One of the ones that I actually do ask, if somebody tells me that they have a dopamine or they have a motivation issue, they're not highly motivated, they're noticing indications of that. One of my follow-up questions is, do you get upset easily? Do you get angered easily? And most of these people will tell you, yeah, I do. I feel like I'm running on edge a little bit more than I think is ideal for me. So that's a pretty good indicator for you on these. And then, given that a lot of us in this room actually are looking at things from a musculoskeletal perspective, the last one up here with tremors, you can think about this in terms of, if you want to look at a far progressed manifestation of this, think about Parkinson's. You know, Parkinson's have the tremor condition. I've actually seen, I've seen it in diagnosed Parkinson's patients where the tremors will pretty much alleviate just increase in dopamine activity. So you can definitely make an impact on that. And then you want to look at this also with your lack of attention. So with the prevalence of things like ADD, ADHD, even decreased mental alertness, this is going to be the pathway in the body, or these are going to be the areas you're going to want to think about here. Now, central dopamine is produced in the substantia nigra, okay? So this area right here, and those projections predominantly go into the frontal lobe. Right? So you may remember back when we were all going through school, and when they got into the neurology portion and they talked about someone having a frontal lobe lesion or some type of accident where they had impact of the frontal lobe and their motivation goes away. This is the reason why most of those projections filter into the frontal lobe. But it all starts at the substantia nigra here. Now this is a pathway that we go through. You start with tyrosine. So tyrosine you would find from, again, protein sources in your diet. And then that tyrosine has to go over to dopa, and then dopamine, norepinephrine, and epinephrine. Okay, so you guys can read the pathway there. But the important part on this, and where I think we have to look at ancillary labs and the importance of having some of these ancillary labs, even something as simple as a blood count with the patient, is if you look at what it takes to to get from just tyrosine down to dopa, you've got to have adequate oxygen delivery and you've got to have adequate iron. So one of the things that I want you to walk away from this, not only from this presentation, but going back and thinking about some of the other ones that we've done, if you look at iron, the general conception about iron is that it's needed for red blood cell production. And that's kind of where it stops at. I mean, nobody really talks about much outside of that. But if you think back through our presentations, we've looked at multiple different areas where iron's incorporated in. Here we're seeing it involved with neurotransmitter metabolism, going from precursors on down to the actual neurotransmitter itself. And if you'll remember when we talked about the detoxification systems, we actually talked about the cytochrome P450 enzymes being iron-dependent enzymes as well. So iron has a much broader activity range than just looking at red blood cell and function and oxygen delivery here. But I digress on this. So important point here is you got to have iron to get from tyrosine to dopa. Now, to get from dopa to dopamine, you've got to have B6. Now, when you think about, let's use our Parkinson's example here. How many of you have had any affiliation with a Parkinson's patient in your practice, right? Okay. Do they put them on dopamine? No, right? You can't get dopamine across the blood-brain barrier. They use things like L-Dopa, you can use carbidopa, these types of medications, right? Well, the reason is and I'll make more emphasis to this later, but you got to use the precursor here to get that across the blood-brain barrier so that that in turn can be converted over to dopamine. But you got to have B6 to make that happen. And again, this is a schematic of looking at how these things begin to play out. So if you're looking at in your axon terminal here, you're going to get the end product production of dopamine, and that gets encapsulated in these vesicles. These vesicles come to this synaptic cleft. They're released in the cleft. They act on the receptor site, and then they get recirculated back into the neuron. And I mentioned the class of medications earlier. These monoamine oxidase inhibitors, they actually prevent the breakdown of these various neurotransmitters, keeping them more active in these clefts. That class of medications is not used a huge amount anymore. I mean, they're still out there. They still do get used, but they've been replaced to a significant degree by other classes of meds. But if you do see something like this pop up, make sure that you're keeping this in mind when you're putting these precursors in. It doesn't mean that you can't use the precursors. I'll give you an example of what I'm saying with this. If you look at the SSRIs, your serotonin-specific reuptake inhibitors, right? Or serotonin selective, whatever. And you're using 5-HTP. You can use 5-HTP. There's concern out there about driving people into what's known as a serotonin storm. If you talk to other clinicians out there and ask them how many times they've seen it, nobody really sees this stuff, right? You can do it, just use reasonable levels to improve the efficacy of the medication, and then once the medication and the supplementation are both doing their job, your patient's stable, then you can wean down the medication and if necessary pull up the supplementation. So it doesn't mean they're not exclusive, you can use them both, just be conscientious about how you're doing that. And then on the back side of this, with that breakdown process, to make that happen efficiently, you've got to have your methyl donors. So we've talked about these, looking at our B12 and our folate predominantly here, but those have to be in play, and you got to have magnesium to make that happen as well. Now, one of the things that you can do, some of the companies out there that look at organic acids, one of the breakdown products that they're going to be looking at is the homovenolinic acid here. That one, you know, if you see that and you're thinking about it in terms of excess dopamine excretion, just take into consideration what you're looking at. A lot of those tests will actually have your methylmalonic acid and what's considered, the nickname on it is feed glue, but it's your folate marker. You know, if you had inadequacies in those methyl donor markers, then the chances that you're going to have an abnormal outcome on your end product or organic acid here is likely as well. So just kind of look at the full picture when you're thinking about it in terms of what is the lab actually showing me about the neurotransmitter stats here. That's why I think if there is definitely one area that I would say symptomatology, clinical manifestation, holds a significant amount of weight, and probably more weight than anything you're going to see on a lab directly targeted to neurotransmitters, this is the area. This is the area you definitely got to be listening to your patients on. Serotonin, social. This is how I think about it. My question here is, and I've had to modify this question a little bit because I used to ask my patients, are you social? And that's, you know, someone will like, well, no, I don't like being around people. Okay, that's not necessarily abnormal, so I had to change my question. Do you find yourself being less social than you have been in the past? Right? Because someone who was never social is probably still not social. Right? So that's how I term that with the patients. Just social interaction there. And you can follow that up with something like, do you have a tendency to want to be a little bit of a recluse? That type of thing. The general nature you're going to think about here, spontaneous, optimistic, cheerful, easygoing, independent, non-committal. It kind of seems like your social butterfly. Right? So if you've got someone that meets that type of presentation, this is probably what their predominance is. Now, someone who's having issues with this, they're not going to find pleasure in very many things. They're going to want to be by themselves. Again, you have to kind of base that on your patient and what their history is there. That may not be an abnormality. And then you'll see I put depression in this one and poor mood. But the truth about this is that when it comes to depression, nearly any of these 4 neurotransmitters in inadequate quantities can be a contributor to depression. So when you see a depressive presentation, don't just automatically assume it's serotonin based. That's just the one that, in general, in our society, we tend to think about it being associated with. But again, any of the 4 core neurotransmitters can be associated with depression. If you look at central production on your serotonin, it's going to be produced in the raphe nuclei, and it's going to circulate back around. Those projections pretty much project all throughout the entire cerebrum there. This is just kind of another viewpoint of it where you're actually looking at a combination of dopamine and your serotonin projections overlaid with each other. Now, if you look at this pathway, you're going to start with, to get the serotonin, you're going to start with your basic proteins. You'll get your tryptophan from that. Your tryptophan is going to go into one that we most commonly know as your 5-HTP, your 5-hydroxytryptophan. And then you get into serotonin, and then ultimately from serotonin you go to melatonin. But the key point in this is the location of some of these various nutrients in here. So now if we're beginning to think about, well, how do I know all this is actually taking place? This takes us back to our micronutrient testing, and the reason we would want to think about micronutrient testing with some of our patients that present with neurotransmitter symptoms. But this is your basic pathway here. And I wanted to make sure we had a diagram here that incorporated serotonin and melatonin, because melatonin has a huge role to play in long-term balancing of the overall 4 core neurotransmitters as well. Your GABA, nervousness. You got somebody who walks in and my question to them is, are you anxious or nervous frequently throughout the day? Or does that happen to you more ideally than you would like for it to? Some of them will even tell you, yeah, I have panic attacks, which is just a logical progression on this. These personality types, these are going to be your more dependable, punctual, kind of level-headed caretaker, team player. These are the people, you know, if you're a team player, they don't really like to rock the boat. They just kind of go status quo. They don't really like shaking things up too much. These personality types, if you get somebody who is that personality type and they have a deficiency, these are the ones that really seem to need the support. I mean, you really got to do something about this category. If you had to pick the one that is probably the most relevant, because if you don't do something about these patients' anxiety or nervousness, you can't grab their attention for anything else you're trying to work with them on. So you really got to support those. You really got to support those GABA regions for them. The anxiety and nervousness is a big one here. Now, this one actually is a little bit different in terms of production, and this is a key point I want to drive home for us here. When you look at GABA originates from glutamine. So if you think back about some of the areas we've talked with regards to glutamine, think about gastrointestinal function, think about the catabolic nature of the body, you can think about immune system function. If the patient has any type of GI deficit, if they got chronic immune stimulation, their ability to produce glutamine or to resource glutamine over to the nervous system away from those other areas that need support is going to be limited, because the body's going to try to preferentially shove it wherever it needs to go to support that area in trouble, right? So those patients might have a little bit of a tendency to be more anxiety types. Now, when you look at this, you're going to see that this goes from glutamine, and then you ultimately drive that into the mitochondria. What happens in the mitochondria? Energy production. Right? That's our energy production locations in the body. And then out of the mitochondria, these neuronal-based mitochondria, from that you get glutamate production, and then the glutamate ultimately converts over to GABA. And then the GABA can have its inhibitory effect at that point. Right? And then ultimately what ends up happening here is that this whole process just goes back into reverse, and you recycle the glutamine levels back up there. Okay? But the point I want to make about this is that this is produced in the neurons throughout the central nervous system from energy production. So if you've got anything that limits energy production at the mitochondria, you're going to limit glutamate activity. Okay? So that's why you've got to, if somebody has anxiety, nervousness activity, you've got to ask them about how are their energy levels, what are the things that are influencing their energy, and diagnose that and work with that, as well as trying to work with the central nervous system aspect of this. There's more I'm going to come back to on that. Acetylcholine, memory. Just straight up ask them, how's your memory? Do you have trouble remembering where you place things? Do you walk into a room and forget why you walked into that room? Is your short-term memory less than ideal as compared to your long-term memory? These are the things that you want to think about. And to some degree, guys, we all kind of get in a little bit of a fast pace at times, and we'll walk into a room and not immediately remember why we walked into that room. But if we kind of stand there and think for a second, we probably can regain real quickly while we're in that area. This, more you're trying to differentiate here, they're just completely absent-minded about why they walked in there. And most of the time, you don't even have to go into that depth of a level of conversation with the patient. Just ask them, how's your memory? Oh, it's poor. It hasn't been as good as it used to be. They'll tell you right away. Okay. These general personality types, these are going to be your artsy, creative people, right? Generally pretty high intelligence level. They're going to be more open to new ideas, intuitive, that creative nature, your inventors, people of that persuasion. And then your deficiency indications, poor memory, forgetfulness, and slowness of movements. Again, if you're thinking with regards to slowness of movements here and musculoskeletal aspects, this is one you want to follow up on. The reason that this comes into play, and the reason that it presents in this manner, is because the neurotransmitter that interacts at the junction or at the synaptic cleft between the nerve and the muscle is acetylcholine. And acetylcholine is predominantly produced from the hippocampal region in the brain. If you guys are not familiar with exactly this, the hippocampus it's a very odd shaped structure. It has almost a horn, like a ram's horns, where it will rotate back on itself. It has that type of structure about it. Just do a quick Google search, just hippocampus, and do some image views on it, and you'll see some pretty interesting 3D pictures on it. I just couldn't find one that I thought was really good for the presentation, but it's worth you knowing about. But this is one of the areas. This is also, if you, where people talk about the impact of stress on memory. I mean, if you think about chronic stress damages memory function, or it impairs memory. The reason is because it's damaging this hippocampal area. And when we use things like phosphatidylserine, we're actually impacting the hippocampus so that we get not only better activity out of the hippocampal region itself with regards to acetylcholine, but we're also resensitizing to some degree the impact that the cortisol has on it. And just another diagram here so you can see how this takes place. I mentioned earlier with regards to B5, your acetyl-CoA. Again, look, this is coming out of the mitochondria here, okay? But you've got to have your acetyl-CoA, and then you bind that up with your choline down here, and then that ultimately gives you acetylcholine, and then it has its postsynaptic activity. Now, the blood-brain barrier. I always think about this when someone is trying to talk to me about the latest and greatest supplement regarding neurotransmitter activity, or how do I assess neurotransmitter activity, these types of things. My first thought on this is I go back to traditional psychiatry. And I ask, what does traditional psychiatry use as their benchmark? Do they have lab tests that they employ to validate which medications they're using? No. It's the most subjective area of traditional medicine, right? Because you just don't get a good read using the labs for neurotransmitters like you do for other areas. It doesn't mean that you can't use the labs to glean neurotransmitter information. Don't misunderstand what I'm saying. You just got to keep in mind that the access to the brain is very limited by virtue of the blood-brain barrier. Now, let me tell you something else that's pretty unique about this. We haven't done this presentation, but we will at some point regarding gastrointestinal function. The same proteins that maintain the integrity of the enterocytes, those proteins that hold those enterocytes together, are the same proteins that hold the cells, that epithelial lining of the blood-brain barrier together. So if you're damaging the tissue in the gastrointestinal tract, if you've developed an immune response against the gastrointestinal tract proteins, you've developed an immune response against the same proteins in the brain, and actually the same proteins in the lung as well. Okay. So if you do have some type of degeneration of the blood-brain barrier, given the degree to which that has taken place, theoretically, could you drive dopamine across? Maybe. Could you drive serotonin across? Maybe. But it's not the most ideal way to try to make that happen. Okay. And that limit is generally regarded by size and the composition of the protein. If there's a positive, negative influence on that, that'll inhibit that passing across that blood-brain barrier. Those are all factors there. But the other thing that is relevant about this, and the reason you've kind of heard me mention central a couple of times here, is that the blood-brain barrier defines the line, it's the division point between your central nervous system and peripheral nervous system. It's also, if you think about things like, I mentioned the substantia nigra and dopamine, and you're thinking epinephrine or norepinephrine, epinephrine production. Well, norepinephrine, epinephrine is also produced from the adrenal glands. You also get your predominant production from the adrenal glands as well, so from that medulla region. So there's a difference between peripheral versus central. Most of your serotonin activity, to a large degree, we think about serotonin with regards to neurological function, but most of the serotonin is in the gut. So there's a difference there. It's the blood-brain barrier. So I've just given you some diagrams here. Actually, I wanted to put this in. If you guys want to really read a good article about this with some good diagrams, this particular article is probably one of the better ones I've found. But just some diagrams here, looking at these endothelial cells, these tight junctions, these proteins that are found in between them here, and then what you allow to transverse those epithelial cells. And then here's another diagram just talking about some of your various pathways, like your paracellular versus your transcellular, and how they actually get across to have an impact on the astrocytes here. So some of your factors that are going to affect neurotransmitter activity. Inflammation. So when you look at histamine, histamine is going to be produced secondary to initiation of the inflammatory process. Histamine acts as an excitatory neurotransmitter, so think about it as being a stimulant. Right? And if you, this is also one of the areas you could go back and look at your blood counts and kind of get some insight on this as well. And think about how these basophils, the mast cells, the monocytes, those are all, the neutrophil elevations, those would all be indications that that patient might have a higher level of histamine release into their tissues. And then what's going to happen there is you're going to get increased permeability of the surrounding tissues, so you want to be able to, they actually open up to allow influx of those white blood cells to go into that area to have some type of systemic impact, or localized, not systemic. To allow neutrophils and monocytes access, control tissue damage, eliminate the pathogen. You guys know how inflammation works here, okay? So I say all that to set the stage for these things. Some of your causes for inflammation: food sensitivities, okay? No, probably not a big surprise to anybody sitting in here. These can cause a problem for us. They can drive the inflammatory process. Pro-inflammatory fats, your refined carbs, adulterated foods, infections, hormones, and autoimmunity. Now, let's look at these individually. So with regards to lack of sleep, here's what's going to happen with regards to lack of sleep. This is a graph showing your blue is going to be your cortisol, and then your red is going to be your melatonin. And what you'll notice is an inverse pattern on these things, right? As cortisol goes up, melatonin's down, and once melatonin's up, cortisol is down. When does cortisol go up? During the day. When does melatonin go up? During the night, right? So they should be inversely related to each other. Now, what you're going to see is that if someone, you can see a couple of things here. You'll notice that sometimes if someone has a high and excessive level of cortisol, especially instead of it dropping off in the later afternoon hours, if it started coming back up, similar to this, you might see that the patient's melatonin levels begin to climb to try to push that melatonin back down. Push-pull, antagonistic relationship, right? What will happen is the more you're trying to produce melatonin, you got to grab that melatonin from somewhere. It's got to be resourced from somewhere. So it's got to pull from serotonin. So you're likely driving down serotonin production to increase melatonin activity, right? As that occurs, the balance between serotonin and dopamine gets lost. And serotonin, even though it's excitatory, in general has more of a calming nature about it. And because you lose that balance between that calming nature of serotonin and the activity of dopamine, the excitability of the patient becomes a little bit higher. So just looking at trying to balance the cortisol, whether you're looking at doing it through improving hypoglycemia, stress reducers in the patient's life, any of those types of things can all influence melatonin production to then in turn further improve serotonin dopamine balance. Okay. Trust me, there will not be a test on this. This is just a diagram to show you how this stuff works. So you got this suprachiasmatic nucleus up here, and then this is just showing, how light-dark cycles influence